Bismillahirrahmanirrahim. Structure and function of nephron. Nephron is the functional unit of the kidney. Each kidney contains approximately 1 million nephrons, and each nephron is capable of forming urine. Nephrons don't have the ability of regeneration. That's why once nephrons are lost with renal injury or renal disease, they can't be replaced. Even with normal aging, there is a gradual decrease in the number of nephrons. After age 40, the number of functional nephrons decreases 10% every 10 years. Thus, a person having age 50 has 10% less functional nephrons. A person with age 60 has 20% less nephrons. A person with age 70 has 30% less nephrons, and so on. However, this decrease is not life-threatening because the remaining nephrons undergo adaptive changes and carry out the normal functions. Let's study the structure of nephron. Each nephron is composed of a capillary bed called the glomerulus or the glomerular capillary tuft. The glomerulus is surrounded by a cup-shaped capsule called the glomerular capsule or the Bowman's capsule. Bowman's capsule and the glomerulus are collectively known as the renal corpuscle. The Bowman's capsule gives rise to tubules which are collectively known as the renal tubules. The first tubule that arises from the Bowman's capsule is called the proximal tubule. It's a convoluted tubule. The proximal tubule leads to a U-shaped tubule called the loop of Henle or the Henle's loop or the nephron loop. The loop of Henle consists of two limbs. One limb descends downward while the other limb ascends upward. The limb that descends downward is called the thin descending limb, while the limb that rises upward is divided into two parts. The lower part is called the thin ascending limb, while the upper part is called the thick ascending limb. The thick ascending limb gives rise to another convoluted tubule known as the distal tubule. The distal tubule leads to a connecting tubule. As its name indicates, connecting tubule connects a nephron to a collecting tubule. The part of collecting tubule found in the cortical region is called the cortical collecting tubule. The cortical collecting tubule receives many nephrons, descends downward and reaches the medullary region. The part of collecting tubule in the medullary region is called the medullary collecting tubule. Many medullary collecting tubules join together to form the collecting duct. Collecting ducts join together to form further larger ducts, and these larger ducts leads to the renal pelvis. We have already studied that pelvis leads to the ureter and ureter leads to the urinary bladder. There are almost 250 larger collecting ducts in each kidney. Each large collecting duct collects urine from almost 4,000 nephrons. Each segment of nephron performs specific functions. The renal corpuscle, that is the glomerulus and the Bowman's capsule, carries out ultrafiltration. The proximal tubule is responsible for selective reabsorption. The nephron loop carries out osmoregulation. Distal tubule is responsible for selective secretion, and the rest of the tubules are involved in osmoregulation. We will study all these functions in detail in a while. There are two types of nephrons based on location: the cortical nephrons and the juxtamedullary nephrons. Almost 70 to 80% of nephrons found in the kidney are cortical nephrons, while only 20 to 30% of nephrons found in the kidney are juxtamedullary nephrons. The glomerulus of cortical nephrons is located in the outer cortical region, while the glomerulus of the juxtamedullary nephrons are located deep in the cortical region, near the medulla. Cortical nephrons have short efferent arteriole that divides into a network of capillaries called the peritubular capillaries, while juxtamedullary nephrons have a long efferent arteriole. This long efferent arteriole extends into the medulla and divides into a network of capillaries called the vasa recta. The vasa recta lie side by side with the loop of Henle. Cortical nephrons have short loop of Henle, while the juxtamedullary nephrons have a long loop of Henle. This long loop of Henle is responsible for the formation of concentrated urine. Function of nephron. Nephrons are responsible for the formation of urine. Urine formation occurs in two steps: the glomerular filtration and the tubular reabsorption and secretion. The first step in urine formation is the glomerular filtration. We have already studied that afferent arteriole brings blood into the glomerulus. The glomerulus gives rise to the efferent arteriole that carries blood away from the glomerulus. The afferent arteriole is wider, while the efferent arteriole is comparatively narrower. As the efferent arteriole is narrower, it creates a high pressure in the glomerulus. Thus, small substances are filtered from the glomerulus into the Bowman's capsule. The high pressure created in the glomerulus is the main force for the filtration process. The fluid that is filtered from the glomerulus into the Bowman's capsule is called the glomerular filtrate. The amount of filtrate produced per minute is called the glomerular filtration rate. About 125 ml of filtrate is produced per minute. However, out of this filtrate, 1% or less than 1% become urine. 99% of the filtrate is reabsorbed into the blood. Small substances like water, glucose, amino acids, mineral salts, and nitrogenous waste products become part of the glomerular filtrate. Large particles like the blood cells, platelets, fates, and proteins are not filtered out. Half of the calcium and most of the fatty acids are bound to the proteins. That's why these are not filtered out. Only that calcium and fatty acids that are not bound to proteins are filtered and becomes part of the glomerular filtrate. Glomerular capillary membrane. The glomerular capillary membrane is composed of three layers: the innermost endothelium, the basement membrane, and the outermost epithelium. The endothelium is composed of holes called fenestrae. These fenestrae are negatively charged. The basement membrane is composed of collagen and proteoglycan fibers. These are also negatively charged. The outermost layer epithelium is also negatively charged. epithelial cells are not continuous, but they are present in the form of foot-like processes called podocytes. These podocytes have gaps between them called the slit pores. As the glomerular capillary membrane is negatively charged throughout, this is the reason that only small and positively charged substances can cross them and becomes part of glomerular filtrate. Proximal tubular reabsorption. Filtrate from the Bowman's capsule passes into the proximal tubule. Almost 65% of the filtered sodium and water, slightly lower percentage of chloride ions, all glucose and all amino acids are absorbed from the proximal tubule into the peritubular capillaries. The epithelial cells of the proximal tubule have extensive brush border surface. This brush border surface increases the surface area for absorption. Epithelial cells also have many mitochondria. That's why these cells are highly metabolic and provide energy for various ongoing processes. Substances from tubular lumen passes through the epithelial cells into the extracellular space and reaches the blood capillaries. Sodium-potassium ATPase pump provides the major force for the reabsorption of substances throughout the proximal tubule. Sodium-potassium pump transports sodium from the tubular epithelial cells into the extracellular spaces. As sodium is transported into the extracellular spaces, that's why there is always low sodium gradient in the epithelial cells. As there is low concentration of sodium in the epithelial cells, that's why sodium from high concentration in the lumen flows towards the low concentration in the epithelial cells. In the first half of the proximal tubule, sodium is co-transported along with glucose into the epithelial cells by sodium-glucose co-transporter. Sodium is also co-transported along with amino acids in the first half of the tubule. In the second half of the tubule, only little glucose and amino acids remain to be absorbed. Therefore, sodium ions are now co-transported along with the chloride ions in the second half of the proximal tubule. Substances from the epithelial cells are transported into the extracellular space. Sodium is transported by sodium-potassium ATPase pump. Glucose is carried by glucose transporter. Amino acids and chloride ions are also transported into the extracellular space. As there is high concentration of chloride ions in the second half of the proximal tubule, some of the chloride ions diffuses directly from the lumen through the intercellular junctions into the interstitial fluid. The transport of sodium into the peritubular capillaries creates an osmotic gradient, due to which water also passes into the peritubular capillaries. Along with reabsorption, secretion also occurs in the proximal tubule. The secretion of hydrogen into the lumen is brought about by counter-transport mechanism. This counter-transport is mediated by a specific protein known as sodium-hydrogen exchanger. By means of this exchanger, sodium is carried into the interior of the cell and hydrogen ions are forced outward in the opposite direction into the tubular lumen. The proximal tubule is an important site for the secretion of organic acids and bases like bile salts, oxalate, urate, and catecholamines. Many of these substances are the end products of metabolism and must be rapidly removed from the body. In addition to the waste products of metabolism, proximal tubule also secretes many harmful drugs and toxins directly and clear these substances from the blood. Proximal tubule is followed by the loop of Henle. Loop of Henle is also called the nephrons loop. Loop of Henle is named after Dr. Henle, who described it first. Loop of Henle is divided into three distinct segments. The first segment is the thin descending limb. The thin descending limb is composed of squamous epithelial cells. These cells have no brush border surface. They also have few mitochondria and show minimal level of metabolic activity. The thin descending limb is permeable to water. Almost 20% of the filtered water is reabsorbed here. This segment is impermeable to solutes. The second segment of loop of Henle is the thin ascending limb. The thin ascending limb is also composed of epithelial cells that have no brush border surface. They also have few mitochondria and show minimal level of metabolic activity. On the contrary to the thin descending segment, thin ascending segment is permeable to solutes, but it is impermeable to water. The third segment of the loop of Henle is the thick ascending segment. It is composed of thick epithelial cells and have high metabolic activity. It carries out active reabsorption of sodium, chloride, and potassium ions. About 25% of the filtered load of sodium, chloride ions, and potassium ions are reabsorbed mostly in the thick ascending limb. Considerable amount of other ions like calcium, magnesium, and bicarbonate ions are also absorbed in the thick ascending limb. Thick ascending segment is also less permeable to water. Let's study the mechanisms how sodium, chloride, and potassium ions are transported from the lumen into the interstitial fluid. An important component of the reabsorption of solutes over here is also the sodium-potassium pump that transports sodium into the interstitial fluid. As sodium ions are transported outward, there is low concentration of sodium in the epithelial cells. Thus, sodium can enter the epithelial cells. Sodium is transported into the epithelial cells by sodium-hydrogen counter-transport mechanism. In this mechanism, sodium moves into the epithelial cells while hydrogen ions are secreted into the lumen. Along with counter-transport, co-transport of sodium also occurs here by means of 1 sodium, 2 chloride, 1 potassium co-transporter protein. As its name indicates, 1 sodium ion, 2 chloride ions, and 1 potassium ions are carried into the epithelial cells. This transporter can be readily blocked by loop diuretics like furosemide, bumetanide, and ethacrynic acid. Loop diuretics block this co-transporter to treat disorders such as hypertension and heart failure, etc. The thick ascending segment leads to the highly convoluted distal tubule. The distal tubule have same reabsorptive characteristics of the thick ascending segment. It also reabsorbs sodium, potassium and chloride ions while it is virtually impermeable to water and urea. In the early distal tubule, absorption of sodium and chloride ions occur by sodium-chloride co-transporter. Approximately 5% of the filtered load of sodium and chloride ions are co-transported in the early distal tubule. Sodium is transported into the interstitial fluid by sodium-potassium ATPase pump, while chloride ion diffuses by means of chloride channels. In order to treat patients with hypertension, thiazide diuretics are used to block sodium chloride co-transporter protein. The late distal tubules and the cortical collecting tubules have two types of cells: the principal cells and the intercalated cells. The principal cells absorb sodium ions, while secretes potassium ions into the lumen. The intercalated cells absorb potassium and bicarbonate ions. These cells in turn secrete hydrogen ions into the lumen. The absorption of water in these tubules is controlled by the concentration of antidiuretic hormone. If more hormone is released, there is more absorption of water by these tubules. As more water is absorbed by the tubules, the urine formed is concentrated. If there is absence of antidiuretic hormone, the tubules become impermeable to water. As no water is absorbed by the tubule, dilute urine is formed. Many cortical tubules join together to form large medullary collecting ducts. The medullary collecting ducts are composed of cuboidal epithelial cells. These cells have few mitochondria. The cells of medullary collecting duct reabsorb less than 10% of the filtered water and sodium ions. These ducts are the final site for urine processing. It determines the final urine output of water and solutes. Sodium ions are actively reabsorbed into the epithelial cells, while the hydrogen ions are secreted into the lumen. The reabsorption of water over here is also controlled by antidiuretic hormone. In the presence of antidiuretic hormone, water is absorbed by the epithelial cells, while in the absence of antidiuretic hormone, water is lost in urine. The tubular filtrate form is finally known as urine, that contains extra water, nitrogenous waste products like urea and uric acid, toxins, and metabolic waste products filtered from the blood. Urine from larger ducts reaches the pelvis. From the pelvis, it passes by peristalsis through the ureter and finally reaches urinary bladder. The bladder is small when it's empty but acquires a round shape when it is filled with urine. The normal capacity of urinary bladder in adult ranges approximately from 300 to 400 microliter. The bladder fills progressively until the tension in its walls rises above a threshold level. The rising tension in the bladder wall above a threshold level elicits a nervous reflex known as the micturition reflex. Micturition reflex is an autonomic spinal cord reflex, but it can be inhibited or facilitated by centers in the brain. This reflex causes either a conscious desire to urinate or empties the urinary bladder. The emptying of urinary bladder is called micturition or urination. We can properly define it as a process by which urinary bladder empties when it is filled with urine. Thank you so much.